Okay, our next speaker is, again, an online presentation. Her name is Roni Rack, Dr. Roni Rack. She holds a PhD in neurobiochemistry from Tel Aviv University and is now leading her own lab at the Volcani Institute with a specific focus on bovine mesenchymal stem cells. Today, Roni will talk about their CRISPR screen, which I'm personally quite excited about to hear, and they assessed proliferation and differentiation capacity. So yeah, Roni, the floor is yours.
Hi, everyone. I was so anticipating to join you in person, but unfortunately the situation in Israel did not allow me to travel. I really hope that time will be more peaceful and I will be able to meet you and see you in person over there and invited to Israel. So let me shift the gear and share with you our results. No, just, okay, perfect. So I wanted to share with you how to use CRISPR in order to study much more about the genetic potential of the cells. So in my work, we will use the CRISPR screen to identify targets that improve proliferation and differentiation of mesenchymal stem cells. So my lab is actually focused on the challenge of making the cells proliferate much faster and while remaining their differentiation ability. So in this screen, we aim to identify targets, genetic targets that enable rapid proliferation without hurting the differentiation efficiency, while allowing the cell also to grow in cost-effective media and implement an industrial scale in culturing methods. So the genetic basis come from, we can look at the genetic basis from multiple perspectives. One of them is just looking at the natural variation of different cow, which is done in my lab by Mamon Abdel-Nadi. But we also can use genetic editing variation. So we can actually manipulate the cells, not only using one genetic manipulation, but using many genetic manipulation and a high throughput screening method, which I will describe immediately. This work is done by Amit Zirman and Ella Samuels in my lab. Amit was supposed to present a poster here, but unfortunately was called to the reserve military service. In addition, in our lab, to enable the genetic analysis and the screen, we also deal with different environmental growth conditions by using different growth factor, different media, and scaffold that introduced to the cell growth. This work done mainly by Orit and Yuval from my lab, and with close help and many collaboration in the cultured meat consortium in Israel, funded by the Science Excellence Foundation. So in mammalian cells, there is a strong dichotomy between cells that proliferate and cells that differentiate. Mammalian organisms have a lot of defense mechanisms that target uncontrolled proliferation, and proliferating cells and differentiated cells are very different from each other, both in mRNA expression and which gene they are expressed. Specifically, we can also see that there is, for example, a different codon usage. So cells that proliferate use different codon usage than differentiated cells. In addition, when there is misregulation of proliferation, the cells actually can become cancer, and it is very devastating for the whole organism. So a lot of efforts are given to prevent uncontrolled proliferation. One cell that is out of the scheme and that is still proliferating in mammalian cells are the stem cells. So, and also in the stem cells round, there is a difference between proliferation and differentiation. So cells that have a high proliferation ability, they usually takes much more effort to differentiate. So for example, embryonic stem cells, they proliferate very fast, but their differentiation potential is, they have an infinite differentiation potential, but it's very hard to make them differentiate. There's a lot of effort need to be done to differentiate them toward the desired cell type. On the contrast, there is the satellite cells or muscle precursor that have a very limited proliferation ability, but they can differentiate much easier to the desired cells. Mesenchymal stem cells are in the middle ground. That's why we chose them as our platform for the screen, because they have a pretty good proliferation potential, but they're not too hard to differentiate. Those mesenchymal stem cells can be produced from different origin, and they have a differentiation potential toward chondrocyte, adipose, and myocyte cells. So we have first isolated the cells from bovine adipose tissues. We characterized their growth, and we saw that we can grow them for a long duration of passages. We can actually grow those mesenchymal stem cells, adipose-derived mesenchymal stem cells, for 25 passages. They have a unique surface marker that let us see that those are actually mesenchymal stem cells, and they have a multipotent differentiation potential, meaning they can differentiate toward several lineages, for example, chondrocyte and adipose tissues. We can see that natural variation can dictate how good the cells can differentiate toward adipose. And we can see that there is different adipose differentiation tendency between cells derived from different tissues, different adipose tissue of the bovine cow. Okay, so next we wanted to employ a CRISPR screen. So what is a CRISPR screen? Instead of knowing in advance which gene we need to mutate in the cell, we want to come without prejudice and try to screen as much cells as possible. So we use the cells we isolate from the bovine adipose tissues, and we want to introduce many gene targets. We grow all of them together as a pool, and only after, without knowing in advance which genes are the best candidates, we can see which genes survive the best. For example, in our assays, which genes proliferate, which gene knockout makes the cell proliferate faster. After we do this assay, we can use next generation sequencing to detect which guide was inserted to the cells. And this is how we can identify which mutation can actually cause the cell to proliferate faster. This is some sort of evolution experiment when we can actually detect without prior knowledge which gene affect the best on the cells. So the first step was to construct the CRISPR knockout library, make a bit of a better selection of the genes target and not knocking out the entire genome. We chose to build on prior knowledge based from cancer. So in cancer, there's a lot of genetic screen done, and leading candidates were already identified to improve cell growth. In addition, we combined it with the expression data of the mesenchymal stem cells. So after this analysis, we could actually choose 3,000 guide RNA, which represents 1,000, approximately 8,000 genes. We chose genes that are that affect the best on cancer cells. So it's actually the opposite from cancer. We want cells to actually grow faster and not, so it's like negative effects on cancer, but it's good for us. So we call them the death map up. So the genes that improve cancer cell growth. We also have a lot of transcription factors in our screen and a specific muscle transcription factor. We construct all of that and put each guide RNA in a specific plasmid, which will target the CRISPR system to create a specific knockout. In addition, we constructed the mesenchymal stem cells with the CRISPR mechanism, machinery itself, meaning the cells that we are using are genetically modified cells. However, when we identify specific knockouts, we can do it without genetic modification and just use a clean gene editing technique. Next, we applied an appropriate selection pressure. So we started with the whole library of genes of knockout bovine mesenchymal stem cells. So each cell has one unique knockout. And we grow them for under normal growing condition for many passages. We can see first we transduce the cells with the Cas9 CRISPR mechanism. After a few days, we transduce the entire guide RNA library to our cells. And then we started following them. And using next generation sequencing in each indicated time, we can actually measure the abundance of each guide RNA, which represents the growth rate of the cell harboring this knockout. And this is how we can see the results, okay? So looking at the y-axis, we can see the change of abundance of each gene. Higher on the y-axis is cells that when we did knockout to a specific gene, they increase their proliferation rate. On the x-axis, we can see how significant was the effect based on 3 to 5 repetition in the library and average of 3 repeats of the experiment. So again, genes on the higher on the y-axis are genes that increase proliferation upon knockout, and genes on the lower y-axis, they decrease proliferation or harm the cells. We can see one specific candidate that increased the proliferation rate significantly in all our experiments. This gene, okay? This gene is quite a known gene. It's p53. It's not very surprising that it came out in our screen. However, it's reassuring that the screen was done properly and this system can be used in our and was designed correctly and can be used further. Another top candidate that appeared was P10. I will discuss more about the P10 results in a minute. Another interesting result was that the cells that harbor the entire library was actually grown much better than the wild-type cells. So although they carry a lot of different mixture of guide RNA and different mixture of cell knockouts, still the population was much more resilient and continued to proliferate toward 125 days, while the wild-type population was already decayed completely. Looking at those cells that continue to proliferate, we can see that a large portion of the guide RNA that remain in the culture was actually p53. So we can see that p53 is quite strong in increasing proliferation rate of bovine mesenchymal stem cells. P10 was also a great candidate. So P10, we generated a single clone of knockout bovine mesenchymal stem cell of P10, and we can actually see that it increased increased the growth rate by 20%. When looking at 20% increase in growth rate, if we can theoretically just grow the cells for 35 days without the P10 knockout, we would get 10 kilogram of cells, while if we actually increase only by 20%, we would get 600 kilogram of cells. However, when measuring the differentiation potential of P10 knockout cells, we can see that we eliminate or decrease the adipogenesis of the cells when we treat them with a differentiation protocol for adipose differentiation. So it is good in increasing growth rate, but the cell does not retain the full differentiation potential. So other interesting candidates coming from the screens, and I will not cover all the genes that we identify, also because it is not published yet, but soon. Other interesting candidate coming from regulation of response to growth factor, TGF-beta pathways, and also pathways involving in chondrogenic differentiation, which actually might inhibit the spontaneous differentiation of the cells, leading to more rapid proliferation. And to conclude this work, CRISPR screen can be used to uncover genes and pathways affecting proliferation rate in bovine mesenchymal stem cells. Genes can be targeted directly, and they can be used to identify small molecules that will be implemented in the protocol of cultured meat growth. P10 and P53 are leading candidates that can be knocked to increase growth rate in bovine mesenchymal stem cells, but might compromise differentiation potential. And using the CRISPR screen platform, we can further identify more genes that involve in differentiation toward myogenic differentiation, or identify genes that are involved in suspension growth and under defined media conditions. And with that, I will say thank you to my lab, Amit, Mamoun, Ella, Yuval, Orit, my great collaboration in the Cultured Meat Consortium, funded by the Israeli Innovation Authority, the support from the Good Food Institute and the Agriculture Agency in Israel. I also take the stage to tell you about a new perspective we have published, and I hope you can read a little bit more in there. That's it.
Thank you very much, Roni. I think we have time for one short question.
Yes. Hi.
Okay. There's a question here. Diana?
I'm not going to struggle.
Hi. Thanks very much for an excellent talk. So can you tell us how many candidate genes did you find in total?
Okay.
Were it a lot or
Yeah,
a few? And did you differentiate the knockout cells into anything? And was that affected? The differentiation?
Okay.
Thank you.
So we identified around 20 targets. We haven't screened for all of them yet. Our thought is to conduct also the differentiation screen. So we actually want to sort the cells using FACS sorting and the specific myogenic reporter to identify genes that can also differentiate. And then we can do the trade-off between proliferation and differentiation. So we will be able, using high throughput technique, to identify which targets improve proliferation without turning the differentiation potential.
Thank you.
So currently we have around 20 candidates that increase proliferation. We don't know yet how many of them actually enable good differentiation, and toward which lineages they retain their differentiation potential.
Okay, thanks.
Thank you very much again. Let's have another round of applause for Dr. Roni Rack.